have both online and offline, and there's many. So also like in people in Avan Lab. So if you use another device, so please switch on your camera so we can have like multiple screens. So I think it will be better for the visibility, right?
So, can you hear us? Yeah.
Okay.
Can you hear us?
Wait a minute.
Yes, yes.
Okay.
We can hear you.
That's perfect. That's perfect.
Recording started.
I will also start the recording just in case. Okay.
I think it's working now. Let's be safe.
Yeah, to be safe. Do you want to start it in a full screen? Oh, it is in a full screen. Because for me it is not. It is not in a full screen in our you shared the wrong thing on your, because in Jitsi you shared the wrong screen. You should share the full screen. No. Use. I will explain how to do it. Just do it here. Okay. Then switch to Jitsi. And then start sharing. Please start sharing the screen, and then you need to, for example, that one is okay. See? That's the right thing. Wait. I need to confirm it. I still have the same thing shared. Do you have or no worries, it's looks like right in case looks like it's okay. Oh yeah, right. So I will try to. Wait a minute. Yeah, I know. I'm just trying to make the recording of the screen. Okay, that's good. And audio. Okay, I started the recording. So, okay, good morning everyone, and good afternoon. Good morning to our Pakistani colleagues, and good morning.
Good afternoon.
Good morning to our Pakistani colleagues, and also good afternoon to everyone who is here in China, including Zixin, who is joining online.
Yeah. Thank you very much. Same to you.
Yeah. So I would like to make sure that whoever is not speaking at the moment on the other side, so please let's mute the microphone so to reduce the noise. Okay. Right, so that's the only thing I would like to just say. And sorry to interrupt you, Fazli. What did you want to say? Fazli?
Yeah, thank you very much. We can see the screen with question and
That is awesome. That is awesome. So I suggest we just give the stage to Verna. She already made the presentation, which everybody had a chance to look to watch online in YouTube. Also, I saw the comments there, and I hope Verna will be able to address them, and we will discuss as always and try to critically assess this paper. Okay, Verna, the stage is yours.
Hi everyone, and thank you for commenting the
YouTube videos, and right now I will address some questions that from this paper. The first question is about PT containing oligonucleotides can be used for antisense effects of in knocking down the genes, but can it be direct effects of the PT containing bacterial plasmid on C. elegans for down-regulating the genes from RNA-seq data? So we know that PT-DNA oligos can indeed be used for the knocking down the genes in some organisms, including C. elegans, and it is designed to hybridize with complementary mRNA sequences and can result in degradation of or inhibition of the certain genes expression at RNA level, and also can be referred to RNAi interference in C. elegans. So PT-DNA oligos can be used to design for the target certain genes of PHS, and when these oligos are introduced to C. elegans, they can be hybridized with target mRNA. And also, they also design some PT containing bacterial plasmid in C. elegans, which and PT-DNA is often present in this bacterial plasmid used for generating transgenic C. elegans genes. And also, but in both cases, the use of PT modifications can improve the stability and persistence of the DNA and RNA molecules in organism cell. But the direct effects of the gene expression is depending on specific designs and purpose of the genetic material. So yeah, so for this question, I think I have introduced. If you have other question, you can ask. For the next question, it asks, what is the significance of using different fluorescence probe and stressor to evaluate the anti-ROS effects of the PT diet? So you use the probe is to for the ROS detections, because there are many different fluorescence probes that can target specific type of ROS, such as superoxide radicals, and using multiple probes will allow the researcher to assess the impact of the PT diet on ROS species and providing some view of the antioxidant effects. And also is for the stressor variability. ROS level can increase due to some stressors, such as heat, toxic, and pathogens. So by subjecting C. elegans to different stressors, we're assessing the ROS level. And also is another significant is for the mechanisms inside. So by studying the RS reduction under some condition can give some insight to the mechanism by which PT diet will exert these antioxidant effects. Also, is relevant to aging and longevity, because RS is linked to many aging and age-related diseases. By evaluating this, the response of the effects of the PT diet on certain type of stressors, and with some different probe, we can assess the potential impact on the lifespan and health span.
Verna, I would like to just add up here a little bit. So for me, it's also interesting. Like, what is the added value of those three different pigments? Yeah. So these probes, like here, one is basically the lipofuscin, right? So the third one, right? Which they use.
Yeah.
Right. Then which is an autofluorescent, which is dependent on the oxidation of lipids and proteins. Very well established, right? And the other two, which is a green and red, you said the red one is more sensitive to, like, what is exactly like the added value? Like, are they kind of like, do they easily substitute each other, or like they also add some additional, like one is more kind of valid for probing some specific aspects of the reactive oxygen species and this type of changes, and the others for the other one, or
the differences.
Kind of, yes. Like, because I think that was the question. Like, why? What is the rationale? It's probably not just to try to show more data, right? But that's of course as well, yeah. But is there some added value? Because I think you mentioned that the red one is kind of more sensitive for certain aspects. Can you just elaborate a little bit more about that?
I need to find the paper. So the first probe is diffuse in the cell and react with some RS species, including hydrogen peroxide.
What? Say it again.
I'm finding the DCM.
Exactly, DCM and this DCF. And you see that they very strangely they make this kind of bars. They didn't really do it very thoroughly in terms of like preparation of images.
So the DCM was more sensitive to hydrogen peroxide and peroxide nitrate.
And what? Hydrogen peroxide and what exactly? What else?
Peroxide nitrate. Nitrite. Peroxide nitrite. PRO.
So that or you can ask Mike to make
the blue.
Peroxide nitrite.
Yeah.
Yeah.
Yeah, peroxide nitrite.
Okay, hydrogen peroxide and peroxide nitrite. Okay, got it. Okay, so that's like more DC. That's for DC, right?
Yeah.
Okay. Okay. So, kind of more sensitive, but I don't see there really much difference. And I mean, they all say, like, all of them, they say the same, and that there is the decline of those markers, right? In the first, fourth, eighth day, but not on the 12th. And I think there was another question. I don't know whether we already discussed it or not, but like, what is the relevance of that? Basically, with aging, those should be actually accumulating, right? So that at that point, they're kind of accumulating, and then they are accumulating both in the this PT treated and non-treated. That's why we don't see the differences anymore. So basically, this kind of change is already beyond repair, right? Is it what they want to say, right? So that on the 12th day is kind of already too late to rescue it by anything, right? Okay, okay, I see. Okay, sorry to interrupt. So you may continue.
And it's also asked why C. elegans is used as experimental model in this study, because also we know that they're using all the wild type C. elegans strands, Bristol and 2, and is the PT-bioengineer and fed with PT-bioengineer E. coli OP50 as control. And they assess the RS level, lipofuscin level, and body and bone size, and all the phenotypic changes, which is easily carried out in C. elegans model, and it's because it has a really short lifespan, and it's really for the cultivation is really easy, and also it has a conservation of the biological processes similar to mammals, and it also really one advantage is that it's genetic how to pronounce it? Simplicity. So it's really well categorized and relative simple genomes, and making really convenient model for the genetic study. So so also we have been asked that what do you think what do you think of translating this study in higher organism, like to explore the effects of PT diet on health and aging of human being? So we need to consider many many things before translating to human beings, such as safety and toxicity. So before the human trial, it's really crucial to assess the potential side effects like adverse reactions, because of a diet containing PT in humans need to be done on the animals model first for the safety assessment, and also there might causing some ethical consideration as well, and also need to check for the biomarkers assessment. So we need to identify the relevant biomarkers for the health and aging in humans to measure the impact of the diet accurately, and that also might cause some oxidative stress. That might include some oxidative stress markers, inflammation markers, and for some disease. So we still need to think about that. And another questions asked that, can you elaborate on how RS level change over time in C. elegans fed with PT containing bacteria, as illustrated in Figure 1? So in Figure 1, we see that the RS level decreased on day 1, day 4, day 8, but not on day 12. But slightly decreased on day 12 with the S2 DCFDA probe, but it's stopped decreasing with the DCM probe. So the implications, I think, is the fluctuations of the RS level changes can imply that first is aging and longevity. So we can see it's relevant to the agents. So sorry. So So we can maybe in later longevity study on day 12, the longevity promoting effects will also be really decreasing. So we can say it's implied to the longevity directly and also implied to the stress response. So it can cope with the oxidative stress and environmental challenge. And also we also imply the cellular dysfunctions because chronic and severe fluctuation in the RS level can disrupt the normal cellular functions, and also it can imply for the inflammations because RS triggers inflammations, and also when this chronic effects having on the health. Also, and the last one I think is it can imply the DNA damage because RS induced DNA damage can lead to mutation and genome instability. And also, I'm asked, what are there some potential drawbacks or side effects associated with the dye-rich PT-DNA? So a dye-rich in the PT-DNA, there might be some potential drawbacks and genome toxicity because it can potentially induce some genome toxic effects, include some DNA damage or mutations, and or some type of immune responses. So that's what I can think of.
Verna, can we stay here for a moment? DCF fluorescent. Have you used it before? DCF?
No.
Or DCM?
No.
No. Only the lipofuscin, right? Okay. For the
when you use the H2 DCF. Oh no, we use the DCF all the
exactly.
time.
Yes, we use DCF and also auto this lipofuscin anyway,
yeah.
right? So, and when you compare like day 1 and like during these several different time points, because you have done that at different time points. Do you remember?
We do day 5.
I remember day 5, but have you ever compared like day 5 and, for example, a different day?
On the elders, you mean?
Kind of,
older.
no, like 5 versus something else. So my question is, what they see here in this increase of the lipofuscin in the blue one, where they don't have any kind of PT. So how kind of does it comply with what you see usually? The blue one,
right?
yeah, lipofuscin as well as DCF.
No, I mean C part.
The green, the blue part, the blue part, kind of 1, 4, 8, 12, like this increase of the of this fluorescence in lipofuscin and DCF. Do you see the same kind of? Is it what you usually see?
You mean it increase?
Yeah. And this kind of slope of increase. Or you didn't check like different time points?
I think it might increase because it's getting older.
Sure, I know, but my question is the slope, whether the slope is like similar to what you used to.
Oh, we have. I think I haven't compared that.
You haven't compared different time points. I see.
But I think it's normal. It's
okay.
It should increase a bit.
Okay, I think we can also talk about the quality of those images later. But let's first go through all of your responses, and so let's continue.
I think I finished.
Oh, you finished already? Okay. So if and by the way, like of course, like as always, everyone can interrupt. So if Nastya or Shanya or Zixin or anyone in the advanced group, if you have any kind of questions just in the middle of Verna's talking or me or anyone else, so you just interrupt and we can discuss. Okay, so but actually Verna finished anyway, right? So that's it, right? So okay, Verna, can you make a final assessment? So kind of a take-home message for you, also take-home message for like the research field. About this paper in general, like what is your take on that, and what is your opinion about, like, in general, the relevance of this study for the whole field? This is the first thing, and second, well, anything else what you want to add? Well, was it a good design, bad design, like relevant experiments, not relevant, or like well done in any way? Okay, well formed manuscript, everything. What do you want to say? So can you make a brief conclusion? Verna?
So I think that this PT-containing diet affects on to reduce the RS level in C. elegans. It has the really good implications for future study. You study maybe in the future it can be used in mammals and for the antisense drugs and to target some biomarkers in human disease and for the treatment, but it will be a long way, I think. Yeah, but yeah, has the potential. And
anything else?
And for the C. elegans research, I think it's quite a new field, and for the bacterial engineering, and also really interesting.
By the way, you didn't see my question there in YouTube, right? Or maybe I posted it too late. So
I think I didn't see it.
You didn't see it. I think I also sent a screenshot as well to you. Anyway, I will ask about that in a moment, but maybe anybody else would like to also ask something?
Well,
yes.
Thank you, Verna. I have. Can you hear me, please?
Yes.
Yes, we can.
Okay. Okay. Thank you, Verna. It was nice discussion. I have a couple of basic queries. That PT-DNA is usually present on the plasmid DNA in bacteria or the chromosomal DNA, and how that gets incorporated into the bacterial DNA. I mean, that is some error prone mechanism, or that is a natural mechanism, and what percentage of PT-DNA is present in the bacterial DNA.
You got the question? Okay, so the question is phosphorothioates are mostly in the genomic DNA of bacteria or in a plasmid? That's the first question. Second, how is it integrated in the physiology? So do bacteria kind of mind? So are they feeling fine, or there is some problem with some physiology? For example, is there some kind of response on are there some mistakes, for example, made by polymerase? Is it kind of error prone or not? So that is the second question. And the third question is, what is the percentage? What is the percentage of this modification in the? I think what Professor Huang was asking, in this particular setting that they use, because they use this this cluster of the operon, I guess, from the salmonella, in order to synthesize this PT. So what is the percentage of this PT modification within the nucleic acid? I think they stated that at the beginning somewhere. Did you see that? So those are the three questions. Fazli, if I misinterpreted it, just let me know, okay?
Yeah, I think that's what I wanted to ask. Thank you for.
Great. Yeah, Fazli, if possible, can you mute whenever you don't speak, mute, because we have some noise, some background noise from your side.
Okay.
Thank you very much. Verna?
So the first question is whether it's in the
Whether it will be in the genome of bacteria or only in plasmid. Oh, yeah.
I think it's in the
What do you think?
um, I think it's in the genome.
I also think so. It should be in the genome, like the plasmid should not be somehow discriminated. I mean, there is no mechanism for that because they use the machinery. So like the only thing is that the substrate is changing, or not the substrate. I think it is being modified in situ, right?
Yeah.
It's not about the substrate. So here I'm not quite sure. And okay, so about the percentage, you found it, right? So they showed in the introduction. They tell about the percentage.
You mean the which percentage? The question is what?
The percentage of this modification among all of the nucleic acid.
Please. For there is one modification percentage, 12 to 14%.
Yeah. So this is not related to this particular model that they used, but it tells us like what we can expect. So we can probably estimate that could be this kind of order of magnitude, right? Nasty, can you stop sharing your screen?
Okay.
So that I can share mine. Do you see my screen shared now? Yeah, you can, right? So yeah, so here they write about this 14%, and the other question was about the so the second question, Verna, do you remember? I already forgot. So one is about the percentage, the other one is about the whether it is in a genome or in the plasmid.
Yeah.
And the third question was, I already forgot. Sorry, Fazli, can you remind us? What is the third one? I'm very sorry.
Right. Basically, I was asking that how that am I audible to you?
Yeah, whether it is.
Can you hear me?
Yes, we can hear you.
Okay. So I was asking that how this PT DNA gets incorporated into whatever that plasmid DNA or the nuclear DNA. Is it like natural process of during DNA replication, or is there any modification in the existing DNA?
Yeah, that's a great question. That's exactly what I was referring to. So this I'm not quite sure. Is it about the substrate which is changing, and then they incorporate it as a usual DNA, or it is done in situ after the DNA is already being synthesized? Do you remember that, Verna? Did you check that? Because I'm not sure how is it done. I'm not familiar. I'm familiar with the phosphothioate modification for different techniques like biotechnological applications of the nucleic acids. Like that is a very well known method. But I'm not familiar about how is it done naturally in bacteria. Verna, do you know?
No.
Is it done? So the question, the answer is we don't know whether it is done kind of before the replication or after. So we frankly don't know that. Okay, I have another question.
Okay, thank you.
Yeah. Okay, thank you.
We can search it collectively in the literature then, for our own understanding later on.
Yeah.
Okay, thank you.
Right. So my question was, which I posted also in YouTube. You remember this this beautiful figure where they have this panel D. You see panel D, right? You see panel D, Verna. And the panel D, they show that actually the survival is drastically decreased upon if they irradiate them with ultraviolet, right? And they don't comment about that. They say, this could not actually increase. Not only it could not increase, it actually diminished. It really diminished the survival. This means that phosphothioate, and then their conclusion is, oh, phosphothioates, they are so great, they are so great. Well, if you expose C. elegans to UV radiation, then phosphothioates are actually bad. So it shows that they really cannot be used as promising agent, because if there are different contexts, it's not only one context, there are many different contexts. And in this particular context, this modification actually drastically reduces the survival. So, and that is a big concern. And they devoted just one sentence.
Why they included this figure?
That's a question. So actually, this point goes like completely opposite to the whole storyline. They included to the main figure, not to the supplement, but to the main figure. And when they have done that, they just commented by one sentence. So in one part, they say, okay, it enhanced survival for like in terms of like stressor, like in case of paraquat and so on and so on, but not by UV radiation. That's what they say. They don't say that it is not only not increased, but it also diminished and drastically diminished. Look at this curve. So this is a big thing. And then later they just devote one sentence to that. I mean, this deserves an explanation, maybe a paragraph. Moreover, this goes in contradiction to these previous studies, because they show that phosphothioates, maybe they are even protective against the UV radiation. And in this case, like it is totally opposite. They are really detrimental. So that's why like it puts their whole kind of concept a little bit in question, in my opinion. Yeah. So that is one thing. Right. So that was my main concern here. And now I think we need to switch to Qiang, because he had a question.
No.
You had it addressed. So you had the same thing, or what was your?
No, I found the answer.
Mm-hmm. Okay, good. Michael, do you have any questions?
No.
Okay, so yeah, so why don't we? I suggest we can just go through the paper again, just a little bit, just to see, like maybe we can find something else. What do you think? So
I think the quality of
the quality
is not.
of yes, exactly. Look at this.
The background is really green. You know, if we want to actually analysis
That is true. I think they use here higher intensity of fluorescence on the right images than on the left one, because the background.
So they increase the
exactly. One can see the background on the right is higher than on the left. This one we can see. So, and one more thing which I see. So they have this scale bar, and instead of just cutting this part from the bottom and just make a new scale bar, they do something strange. I have never seen something like that, like 0 to 250 with the arrows. Here, look at this. What is this? Like this pixelated scale bar, and they put this kind of, it's like, instead of just spending like 1 hour to make a good figure, they just, spend 5 minutes and say, that will be fine. We just send it to communications biology. They don't deserve more. Like it's kind of shows that they don't care much. That is very sloppy, and it is ugly. So and they have this everywhere. You see, they have this scale bar here again. What do you have this pixelated thing?
Because they are from our college.
They are from our college. Okay.
And I
really?
Don't see our machine have this type of scale bar, so they maybe just.
I mean, in every.
They just draw.
In every. No, I don't think so. Like here, that one, which is probably embedded, they just didn't know how to get rid of that.
Okay.
So, and even there in our college or in any other college, I will be eager to tell that again and again. So, dear co-authors, dear authors, so I just wanted to say that, I mean, it's better to care about like what you do. So you just try to make it in the best possible quality. So if you acquire an image, make sure that they acquire it in a TIFF, so in the like highest possible resolution, and then without the scale bar, because that's possible just to acquire without the scale bar, and then later also one image with the scale bar, and then you can put the scale bar on top of that, so it's nice and neat, and you don't need all these kind of empty spaces, you see. So you could have they could have positioned it in a way that kind of to reduce this blackness. Here it looks like they needed more data to fill in, like to make more figures, right? I mean, okay. So, that's just a little bit about this way how they arranged it. Here. They never write any significances. They write significances only in the figure legend. I don't like it. I prefer that the figures would be read already immediately from the image, from the figures. So you don't need to even to go to the figure legend, so that they can be kind of self-speaking. Yeah, so that is much easier to interpret. Same here, right? No significances included. Like it's so easy just to put the significances on top, right? Here, why do they include this FPKM data? I mean, does it contribute any way to the story? They just fill in the empty space. They just want to just add up a little bit so that the figure is a little bit like bigger.
Maybe we don't know their purpose because they are microbiologists.
Still, I mean, like what does this panel A tell us? Does it contribute to the story? Not at all. If you really,
they want to compare.
They want to show that they can do sequencing. No, they want to show that they can do sequencing. Really, this is the message of this. We can do sequencing well. That's it. So that's the message. So I really I don't understand. If they want to include it to the supplement. Actually, I would not even include it to the supplement because it is diluting the whole point and the whole message, right? Because you want your message to be as strong as possible. And anyway, for the sequencing data in all reputable journals, they require the full data to be uploaded as the data. So this is not necessary at all. So this is one thing. Second, so they do this S plus at the day 4. Versus different time points, like day 4, day 12, day day 12, day 4, right? And then they compare S plus versus day 12, S plus versus day 4. Oh, I mean S plus at day 12, S plus and at day, all right, okay, and then they compare. This is really strange way to do this. So instead of going for like one specific day, they're going for like overlap of whatever upregulated genes were at day 4, and they stayed upregulated at day 12. That's their message here. Same here for the downregulated ones. So whatever was upregulated at downregulated at day 4, it remained downregulated day 12. Why they did it this way, I'm not quite sure. So firstly, at day 12, we don't see much difference in the in this reactive oxygen species anymore. Secondly, like does the hypothesis imply that whatever is relevant for this particular phosphothioate should remain and should be like persistent, like from day 4 to day 12? If it is the hypothesis, then how did they come with this hypothesis and why? I didn't see the explanation. So I don't quite understand this kind of logic, why exactly they did it. What I suspect, what I think, is that if they would do it just like that, just day 4 against day 4, or day 12 against day 12, they would have too many. Mm. So they decided, okay, by that we can just okay, make an overlap. But I have a problem with that, because if they have 3 samples per group, and that could be the natural variation as well. Like they can also pick up some kind of irrelevant things as well here. So I'm not quite sure like that is the right way to do this. So that's questionable, really questionable. Okay, and then they find here some kind of high upregulated, high downregulated ones. I guess that is related to this overlap, this 234 and 115, I guess. And then they say that, okay, there are some examples. Okay, they provide some examples, but they just don't go into that. So they don't go into the mechanism much. They just show correlations. They just show, okay, this is up, there are some pathways which are up, down. Then they do something even more strange thing. They go S plus D12 against D4, S minus D12 against SD4, S plus D4. So this cyan, right? This 859, this one is this non-phosphothioates. So like how they age basically. So it is the overlap of how they age versus the aged phosphothioate against the young non-phosphothioates, and then young against young. This logic I don't understand at all. Do you see this overlap? I think here they made the overlap just to reduce the genes, just to compare like whatever with. I don't really see the hypothesis. How can you do this? How can you make this kind of strange entities? How can you compare in 2 times? Firstly, you have the same group against which they compare. They compare the young non-phosphothioate oligos, right? Non-phosphothioate group, right? This S minus D4 in all 4 cases, okay? And then they compare phosphothioate old one versus the young, as well as the young one here, and this cyan. This is the strangest of all. So, and clearly it is the same sequencing data. So basically they analyze it twice. They took the same data, they make one kind of overlap. Like young against young. Upregulated, no, that is upregulated young and upregulated old, and kind of overlap of that. And here is downregulated young and old, okay? This is already strange, okay? But they don't stop here. They just, okay, they just made the analysis based on that. Then they say, okay, what kind of else data we have? We don't want to go into like loss of function or gain of function. We just analyze this data again. We just take the same sequencing again and we do this. And then they come up with this. They just, as a basis, they take the young non-phosphorothioate and compare with them 3 other groups. This is biologically, like, it doesn't make sense, really. How can you compare 2 entities which are the phosphorothioate against non-phosphorothioate, the yellow, and the purple? These are phosphorothioate against non-phosphorothioate. But the cyan group is the non-phosphorothioate against non-phosphorothioate, and you compare them all against each other. Why? Why do you want to compare these entities and just find the overlap? What is the logic? What is the sense here? This is totally not. I don't know how one can do this. I really don't understand. So, which means that whatever they produce there, like we cannot. For me, at least, I don't even need to look into that because it doesn't make sense. These data don't make sense for me at all because the initial settings are wrong. And then, okay, and then they show this. So what does this figure, what does this panel tell us? Panel C. Panel C.
Why do you think that the setting is wrong?
Why the setting is wrong? Okay. There are 2 different groups overall. Like your study, you study phosphorothioate and non-phosphorothioate. Okay. Okay. In each of those groups, you have subgroups: young phosphorothioate, old phosphorothioate, and here young non-phosphorothioate, old phosphorothioate. Yes. So 2 different groups. What they did first is they checked young against young and old against old, and then made an overlap. This, at least I understand the logic. And I say, okay, they look for those which are upregulated in young and remained upregulated in old. That is the logic. This is already questionable because, okay, in this regard, they imply that everything what is relevant for the mechanism of the phosphothioate protective action are those genes which must remain changed from the young to the old. This is questionable. Maybe this protection is important only during the young but not during the old, or vice versa. Maybe it's important during the old but not during the young. How do they know that it is about the persistency? So I suspect they did it just to decrease the number of targets in order to make this whole analysis easier. That's what I think. Maybe I'm wrong, but I didn't see the explanation. How did they come to this hypothesis? But the second screening, firstly, it is just the same data. Basically, you have the same sequencing data, the same 4 groups, but they just analyze it differently. So they take as a reference point the young non-phosphorothioate group and compare all 3 others against it. Like you have non-phosphorothioate, young and old, and phosphorothioate, young and old. They take young non-phosphorothioate as the one reference point and compare all 3 others against each other. So 3 different settings. Those are 3 different circles. And then what they do is they overlap them. what does it biologically mean that you compare, for example, this Qiang group, which is non-phosphorothioate against non-phosphorothioate, basically those genes which are changing with aging in non-phosphorothioate animals. What does it mean?
I don't know why they use non-phosphorothioate day 4 and compared to
you see, you start to understand. You start to understand. Yeah, that is the point. Because if you just you know, if you don't go much into the detail, kind of looks solid, right? They have done so much. They have so nice FPKM. Look at this. They can do sequencing, right? And then in the end, you look precisely and you understand that this kind of overlap, it does make sense.
Or maybe we just didn't get their purpose, or maybe
no, it doesn't have okay, the first one I could understand if they would write, we think that those genes which could be relevant and they are important for this protective action of this phosphothioate, they must be retained from the young until the adulthood, and this change must be retained. I would say, okay, based on what? If they can reference and they say, okay, here in the studies they say that, okay, maybe I can buy it, but still I would be kind of concerned. But they don't write about that. I think, I'm not sure the authors, but I think that they just use it in order to reduce the number of studies. Instead of having 2,000, they now have 100, so they can show nicely 100 which are hit. But what are these hits? That's the question. So the initial setting is wrong. But when they go again, so you take the data again, and they don't bother to generate some data again. They just say, okay, just reanalyze the same data again, and we show them again. But can we show the same data again? They say, no, it's fine. We just reanalyze it, so it will be different data. It will look differently. It's the same data. And it is done in the most, like, I don't know, of course you can do it even worse. But this kind of setting, you understand me. Like, this is irrelevant. I don't even need to look into this. I don't need even to look into this, because of course they can do, with so much data, they can find some peaks, something up, something down, but it doesn't make sense. All of it doesn't make sense. Just forget about that. If you see something like that, if you see this kind of setting, you understand the authors don't know what they do. And this is communications biology, ladies and gentlemen, yeah? So this is kind of research we are dealing with. And it happens also like in top journals, but this is really outrageous, I should say. Okay, look at the panel C. What does it tell us? Panel C.
It looks messy.
No, it looks solid. It looks like they can do heat maps. Yeah, right? But what does it mean?
Even indicate the genes clustered.
Yeah, what does it mean? Okay, it can cluster. It can cluster into 4 different clusters. Okay. And even the genes are kind of clustered.
Yeah,
okay.
It's really strange.
No, they can do clustering analysis. That's what they say. They can do heat maps and clustering analysis. That's what they can do. That is the research we're dealing with, yeah. And why is it here? Yeah, it doesn't have any purpose. There is no indication. Okay, some pathways, whatever. No, nothing. It just stays there to fill the gap, to fill the blank space, to make the figure bigger. That's it, really. And then okay, they do some kind of.
Okay, they have the anti-space of.
Sorry.
You see, they have the anti-space because it's supposed to be 6. So they have the anti-space here, so they want to fill it.
Exactly, I told you, yeah, of course. Fold change, there is a space missing, fold change. Sloppy, you can see it is sloppy, like from the beginning to the end, like whatever you take, it is very sloppy. and then they are trying to make some
the genes changes not really. You see, there are a lot of genes that didn't have any change.
Firstly, don't even look into that. This data is crap. Okay, this doesn't make sense. Like those genes are, which they are showing in D, E, F, G, H. Those are from those overlaps, 185 and 218. I assume. And if it is the case, you just don't even need to look into that. If it is something not related to them, then we can look into that, of course. Do you know whether these D, E, F, G, H are from whole change heat maps of the genes for ROSCA S minus D4 samples? Okay, so let's imagine that D, E, F, G, H doesn't have anything to do with this overlap, and this is kind of unbiased. Then we can look into that. Okay, so okay, let's look into that. Okay, there is something going up there, then something is going down, like SOD4, right? Then HSP 12.6 is also going up, and then it's going down. And okay, but it is going down in the non-phosphorothioate, but it stays up in both the young and old phosphorothioate. Okay, so okay. So basically, we need to see those double hits. Yeah. So like this GST20, this GST20 has like a double hit in this phosphorothioate with young and old. Also GST18. So okay, there is some upregulation of the glutathione S transferase, which can suggest that there is some kind of increase in the antioxidative genes, but that is just a correlation. Yeah, this is nothing about mechanism. They don't even show any kind of causation effect, no loss of function or gain of function, right? So like there is nothing about the mechanism here. They just imply this. All are just correlations. Then they go for some kind of messy stuff, kind of modern system biology, showing those different interactions, which doesn't, for me, doesn't make sense at all. But it takes place, and also like yeah, it can be used as a figure 6. You see, so they have figure 6. Or maybe Verna, can you explain to me like what do they mean by figure 6? What is the take-home message? Or anybody, Michael, can you explain to me what does it mean? I also cannot explain this. So how did they come to the cell cycle and pseudopodium? Why did they mark them as red, but not others? Do we understand this from this? No.
But they wrote a lot of analysis.
Yeah, they can do sequencing. They can do analysis. Great. That is great. But you don't publish good papers if you just can do sequencing and good analysis. Like there should be something more behind. There should be hypothesis driven or whatever. Okay. Then they have some examples of like really hugely changed. Changed pathways. This is all correlative, and they go here for something which is highly upregulated. Whether it is from the overlap or not, I don't know. If it is from the overlap, then I would just outright say that I don't look into that. And then they say that the top upregulated is something to do with neuroactive ligand receptor interaction, and the downregulated is from DNA replication. Okay, so that might make sense, right? So phosphothioates, they are usually used by bacteria in order to evade the restrictases. So their own restrictases are used in order to target the viral genomes, but they use phosphothioates in order to protect their own genome from these restrictases. So that is one of the function of it. So it might be also relevant for the replication here. But this is, it looks like it's all correlative. I didn't know how they even come to this figure 7. So they don't write. Do they write whether figure 7 is related to these previous overlaps that they found, Verna? Because the overlaps are the worst what they have. And then in the end, they say, okay, the conclusion that there is yin-yang mechanism, whatever it means here, about the reactive oxygen species inhibition by these phosphothioates, and then they show some kind of redox neuroactivity, sulfur metabolisms, which we don't see in their supported by the data. At least I see only correlation. I don't see any kind of causation. Well, like my attitude, as you may probably understand, and as you may see, I don't understand how can the editors of communication biology let it through. That is not nice at all. Totally descriptive, and some of the points are also like really questionable in terms of the methodology and also interpretation, which is I mean, if you do not great method, but you don't misinterpret it, you just say, okay, that's a weak method, but we think still that's one thing. But if they overinterpret based on some kind of really questionable analysis, that's a totally different thing. So yeah, I'm really upset about that. So that is my take. So would anybody like to add here, or maybe maybe we can discuss, maybe someone has a different opinion? Qiang or Nastya or Verna?
Mike?
Zixin or anyone in Fazli's group?
So the whole point is that they want to show us that they can do what they think they can do, or they even can do.
No, they need to publish.
Yeah. And
They need to publish, and they just show some descriptive data, but they don't go much into the mechanism. They don't show how. They show, okay, that happens, but that's it. It's not more than that. And even that is not done well, because we found some kind of mistakes, logical mistakes here. Anyone? Ayman, Kuma, or anybody else? Fadli, any comments here?
Well, Ilya, we have thoroughly enjoyed your comments. These were wonderful and rigorous about the paper, and my students, they have learned a lot from this discussion. And thanks to Verna, she has taken this paper for discussion. Not always all the good papers should be discussed, but the papers with problems should also be discussed so that they can understand what the floppy experiments are and how the data integrity means to understand the biological mechanism. So your insight was great in making this important point, how to look into data and how to design experiments for really good science. Thank you very much.
Yes, I agree. I also think that it makes sense also to discuss some not so good papers to understand and also to find the flaws, because sometimes you don't find the flaws so easily, as we saw in some of our previous discussions. And yes, I agree. So Verna, thank you for the nice presentation and for answering the questions. Yeah, Fazli, by the way, are we going to meet in 2 weeks, or like what is the plan? Like, it is up to you whether we make the final session or not. So what do you think?
Well, I can be available, but I guess you might be going on holidays, so we can postpone it for another week, as I mean that is convenient to you.
Well,
And
I think
second week of January, I think, or whatever is feasible in January.
yeah. So according to our plan, we planned this last session. So basically, we can keep it on the schedule. So if you want, then we can make this final session in 2 weeks, and we will be of course happy to discuss it again. So that would be
Yes,
great.
yes, we can be available in like first week
And
of January.
I would like to thank everyone for the contribution, for the nice discussion. Also, like the people from my lab, as well as from Professor Awan's lab. So I think we had a really interesting and nice discussion, and very useful. And yes, then we will continue in 2 weeks. And I wish everyone nice, happy, Merry Christmas and Happy New Year. And wish everyone here also a nice day.
You too have Happy New Year in advance.
Thank you.
Thank you.
Thank you. Bye bye.
Bye bye.